Hello, I'm Joe Beechem. I lead R&D for NanoString, and I'm going to talk to you today about a topic I get asked about a lot. What about all these cosmic sensitivity comparison studies that are done? So this will be a short, deep dive on cosmic sensitivity comparisons. I'm just talking about our subcellular high-plex imager, our cosmic spatial molecular imager. It seems like there's some confusion in the field. I don't know exactly where it comes from, but from the very beginning on CosMx design, we had one design goal: recapitulate all the capabilities of single-cell RNA-seq, but with subcellular spatial resolution. So we don't have to invent a new metric to measure sensitivity. We use the great metric that single-cell RNA-seq came up with. Sensitivity is really simple. It's the maximum number of transcripts you get per single cell. And it's also, there's two terms. You got to get the maximum number of transcripts per cell, and you want the largest diversity of different genes per cell. That's sensitivity. That's all there is to it. And that's what I'm going to be emphasizing today. And I think whenever you hear a sensitivity number, remember, you should be asking for transcripts per cell and genes per cell. That's it. That's really the only spec that really captures sensitivity in a molecular imager. Now, there's been a number of studies done trying to compare different platform sensitivities, and they really are intrinsically flawed. There was one on the left you can see was complete N=1 study. It's like you cannot compare a bunch of platforms with an N=1 study. But that's what's out there, and that's where they had sensitivity measurements against. There's also a study on the right, and the study on the right was really intrinsically flawed because the start dates of the measurements were all different. None of the CosMx measurements were made for 44 days until 44 days after sectioning. Poor Merscope had one at 74 days, and you really cannot do a systematic comparison of platforms on either an N=1 study or when all the start times are different, because that really makes a huge difference to the quality of the data coming out. So your comparisons have nothing to do with the platforms. They have to do with when they were cut. So let's start taking a look at cases where they were cut at similar times on similar tissues. I'll start with the very first one that came out. This was a case, and there was some debate on whether universal panels like we launched at 1,000-plex were going to be able to compete with a organ-specific, disease-specific, lower plex panel. So this is why we're comparing the Xenium 280 breast cancer panel on breast cancer tissue versus CosMx 1,000-plex universal panel. And this study was done. And what does the sensitivity look like? Here you can see what the sensitivity, basically, CosMx is going to see about 5 times as more transcripts per cell as the Xenium, and they see about 4 times the gene diversity on a per cell basis. So the universal panel, in terms of net sensitivity, obviously is higher sensitivity than an organ-specific panel that was examined in this case. But that was, and then if you looked at it in detail, so look at just the overlapping genes, say, and in the tumor bed, you see roughly commensurate sensitivity in CosMx and Xenium, slopes near 1. And then if you're in the stromal region, it looks like the Xenium was a little bit higher. But there was a clue why it was higher by the inset, which shows differences in cell sizes between the cell sizes, segmentation sizes between the two platforms. And here's the segmentation in the stromal area. CosMx on the left with data-driven protein membrane based segmenter, and the Xenium with DAPI nuclear expansion. And that was the essential origin of the sensitivity difference. And at this time, I believe the Xenium platform is actually trying to have real data-driven segmentation now. But the initial pattern was the same, is that more transcripts, more diversity of transcripts seen by CosMx. So now let's look at an expanded, well-controlled study, and you can watch the talk for the majority of this data here on this slide. But let's take 16 blocks, 11 patients. Let's cut them at the same time, same thickness. One goes to a center of excellence for Xenium, the other one goes to a center of excellence on CosMx. And let's see what's measured. Okay, so let's go to simple stats. What were the mean transcripts per cell? About 4 times the number of mean transcripts per cell and about 6 times the gene diversity per cell when examined in this really rigorous manner. This is very indicative. I go back and reference back to the original one we did with Yutaka Suzuki, very similar type of replication. These 16 blocks really fit in very well with the tumor block we examined with Yutaka. Now, if you do this and examine only the overlapping genes, it looks like we're even more sensitive than the xenium on this particular study, but they're very similar, commensurate. But I think the key thing is look at those violin plots over on the right-hand side of the total genes, the transcripts per cell and the genes per cell. And you can see the CosMx violin plots in green, and you got the xenium violin plots in blue. And I challenge anybody, go show that data to your bioanalyst, your data analyst, and say, yeah, which data set do you want to try to get information out of? I imagine all your data analytics people will say, I'll take the green violin plot over the blue one. Now, one thing that Parent Beal was able to find, since he's got so many blocks, he measured the DV200 of those block sets, and what he saw was that the xenium performance goes down as the DV200 scores go under 40. And that's roughly about half the hospital samples if you follow the work in the literature. So xenium will have difficulty. CosMx, which was designed for sort of real world FFP samples from the beginning, has less dependency on the DV200. So these are all sort of high-plex. CosMx always been high-plex, and xenium's sort of always been on the low-plex side. Xenium has now recently launched the Xenium Prime. It's a 5,000 plex. So now we can start doing some comparisons of 6,000s versus 5,000 plex. So we don't have this big discrepancy in plex. Now, to accomplish that in the Xenium, they actually had to add additional steps to the overall workflow before you actually put the high-plex Xenium samples onto the instrument. And I'm just emphasizing the difference. CosMx, it's identical. It doesn't matter if you're doing 1,000 plex or 19,000 plex. It's exactly the same sample prep. You just have a sample prep, hybridization, you do your protein-based imaging segmentation markers, and you go right into the readout. You can see on high-plex Xenium, got sample prep, but then there's actually 3 additional steps that are unique to the 5,000 plex, additional primer hybridizations, RNase, polishing, and you got another hybridization, then you ligate, then your rolling circle amplify, and then your segmentation comes at the end. And all those steps with the little arrows means you got to really, those are hands-on things. You got to accomplish one and accomplish another. So the workflow up in front of high-plex Xenium has actually got, as you can see, reasonably complicated there. These are all individual person's hands working on this before it actually goes into the instrument. So that's one of the important things, high-plex chemistry differences. It really makes a difference on what chemistry you're using. In CosMx, all of our spots, if you actually think about what you're imaging, high-plex image is defined by the spots of the individual transcripts and the fluorescence intensities of the original transcripts. For CosMx, it's identical. We make them in the factory. They're all 20 nanometers in size. They all have exactly 30 dyes. They're manufacturing by the molds, so it's all under GMP. Whereas you think about the way the Xenium works, you're literally, every lab tech, whoever's doing that experiment, basically making the detection reagent one at a time on the tissue. Now, one thing you'll find in the literature is when you're doing rolling circle amplification, you can read all the spots don't amplify at the same size and at the same rate. And as this was highlighted in the literature here, due to the complex environment of crowded proteins and other biomolecules, rolling circle amplification is not as uniform as an in vitro lab experiment, resulting in amplicons with a relatively wide distribution of their incidence intensity. So RCA is always going to have, you can see there on fixed cells, which are almost an ideal case, the fluorescence intensity of the different amplified spots are going to be like something on the order of 3 times different from each other. So now we can look at high-plex versus high-plex. So this is a nice controlled case again, colon tissue, serial sections right at the same time. They go to, this is Pam Beer July's work again, goes to the Center of Excellence Xenium, goes, and then the other slices go to the CosMx SMI, And now what are that sensitivity differences you see? And what you see now is the mean transcripts for these high-plex comparisons. The CosMx is still, it's up about 3.5 times as many transcripts, about 3 times as many unique genes. If you look at only the overlapping genes, and it turns out there's 2,550 of them, there's actually, instead of being commensurate, now Xenium or the CosMx is about 2 times more sensitive on those 2,550 overlapping genes. And similarly, mean unique genes overlapping about twice as many of them. So as you go up in plex, due to all the complications associated with rolling circle amplification, our chemistry stays identical. You'll either get a broader separation between the CosMx sensitivity and the Xenium sensitivity. We are going to be better and better as you go to higher and higher plex by design. And one thing also, if you compare the 6K and the 5K panels, you can see that we got basically 23% more targets. That's our plex differential. But you can see the way we design these panels. We've been designing panels at nCounter for like 15 years. We're pretty good at doing this. You can see that you get 84% more ligand receptor interactions you can do with the 6K. And you can see the reactome coverage, 81% more pathway coverage that you get out of those panels, sort of just from the intrinsic design of the panel. Okay, but we're not limited at 6K. I'm not really even pushing the limits of what SMI can do. We're all the way at 19,000 plex now. And now you're really getting to the place where you can actually literally superpose our data onto single nuke RNA-seq, which you see over on the right-hand side. We're seeing very similar transcripts per cell, genes per cell. We're over 1,000 transcripts per cell. In this sample, I measured over 1.4 billion transcripts. So now you're at the place where CosMx is essentially very commensurate with full-blown amplified single-cell RNA-seq, except we got the XYZ coordinates of everything. So I think, this is a controversial area. I think in the end, publications have the last word, and I just grabbed a couple of recent ones. So, we're over 100 publications and reprints are in there. These are a couple recent ones. We had a Nature paper June 5th. Another Nature Genetics one came out just a couple of days ago, July 27. I actually got informed there's 2 new Nature Genetic papers that have been accepted for CosMx. So when you look at these, and you look at this data and the sensitivity and the descriptions of CosMx, again, you just go back to the literature. You do not get papers in Nature and Nature Genetics if you got false discoveries happening all over there. It's just not the way it works. So here's the summary. Remember, sensitivity, just transcripts per cell, genes per cell, okay? A couple of really well-done control studies now been done at Northwestern University. And you can see when it's high-plex, CosMx, low-plex, okay, we get still higher sensitivity numbers, overlapping genes, essentially commensurate. And if you're looking at the lower right, now we got high-plex to high-plex, the discrimination is even larger. Sensitivity increases on CosMx were about 3.5 times, more transcripts per cell. And then the overlapping genes, the differential is even wider. So we're almost twice as sensitive as high-plex Xenium. So yeah, hopefully you'll be able to sift through all the noise that are out there. Just remember, compare the single cell RNA-seq and just remember transcripts per cell, genes per cell. And I think the sensitivity differentials are pretty clear on these well-controlled studies. Hey, good luck on your research and good luck on evaluating spatial platforms.